Hello, and welcome back to Beyond Networks, the evolution of living systems. This is the core module of the course, and I'm really excited about it. We're going to talk about the ultimate systems, the main sort of target of any biological research, and that is the organism. And we're going to start this module with a lecture about the simplest organisms that we know of, and those are single cells. And the lecture, as you may have noticed, is entitled "The Cell is Not a Machine." More precisely, what we want to look at is how this toolkit that we've built up over the rest of the course so far, this basic recipe for mechanistic explanation, how and if it applies to explaining the function, the behavior, the entire organism. How does it work? So we said the dynamic mechanistic explanation consists of two main steps, right? Very basically, you have mechanistic decomposition. You can do genetics, identify the parts of the system, and then localize function to those parts. So you want the components and their activities. You can do causal modeling to look at the interactions between those parts, the causal structure. But then you have to recompose the system to see how those activities and interactions at the level of the components orchestrate to generate the overall behavior of the system. And I've shown you with the gap gene system that this works very well for sort of subsystems of a living organism. But does it apply to the whole? And that's a very, very difficult and very central question in this course. So some people think yes. So if you do a lot of this, then you will just understand how life works. Here is a textbook that I found, The Cell as a Machine by Michael Sheets and Henry Ryu. Has a bunch of really interesting quotations in the preface. So Sheets is the director and founder of the Mechanobiology Institute in Singapore. So what is mechanobiology? They say, the premise of this book is that cells are small, self-replicating machines that exploit fundamental physical and physical-chemical principles of mesoscale objects, the larger objects, to pass on the DNA of the organism that they form. The only point of a cell is to pass on its DNA, and it's basically a small machine, self-replicating machine. We don't usually build self-replicating machines. I don't know how a self-replicating machine works, but let's see. They have more. Our approach is to model the cell as a complex machine that has been selected for robustness over many millions of years. As an evolutionary biologist, I know that you cannot select for robustness because it's a dispositional character trait, right? It's not something an individual has. It's how you react to changing circumstances, or how many of your offspring are going to be viable. So you can't select directly for that. There's a lot of work on that we'll visit later on. Okay, so the cell is just a vehicle for DNA. We select for robustness. Oh, and here comes, of course, the core of this approach: to understand and control both biological and biochemical aspects of cellular functions. That is the point of mechanobiology. By treating cells as machines, we can have the illusion that we can control biological systems like we can control a mechanical system. I love this car, by the way. Why a car? It's going to come back later in the lecture. So the cell car here is not a very good metaphor, okay, for cells, but it's very popular. It's called the machine conception of the cell. And the whole lecture that I'm going to give today is based on an absolutely wonderful paper by philosopher Dan Nicholson, currently here in Vienna at the Konrad Lorenz Institute, moving soon, George Mason University. And so he's written this paper last year that sums up the whole argument in a lot of beautiful detail. I highly recommend you read this. So I'm going to summarize Dan's argument here. And so he starts sort of describing this traditional machine conception of the cell. It goes back to various people, but here's an example: Jacques Monod, in his famous Chance and Necessity. It's a great book. It's one of these great books that makes a wrong argument very clearly. I highly recommend it, no kidding, that you read it, because it's really interesting, and it shows you very clearly where, you know, Monod's thinking goes wrong. Basically, he writes in that book, through its properties, by the microscopic clockwork function, beautiful, that establishes between DNA and protein. Yep. As between organism and medium. What? So extrapolation, DNA makes protein, organism creates its environment. I don't understand. An entirely one-way relationship. Here's the central dogma of molecular biology. This system obviously defies any dialectical description. There's no mutual interactions here. It is not Hegelian at all, but thoroughly Cartesian, of course. A Frenchman would say that. The cell is indeed a machine, he says, cynically looking, smoking in his lab. Okay, so this is the sort of view we're dealing with here. And it's still very much with us, okay? So the basic principles of this view are, the cell is basically an intricate piece of machinery whose organization reflects a pre-existing design. So there's sort of a blueprint of the cell in the genome. Cellular structure is wholly intelligible in reductionistic terms. So you understand the genetic interactions, you understand the structure of the cell. A cell's operation is governed by their deterministic laws producing predictable, controllable behavior, because that's what machines do. Okay, so here's a representation of this. So networks are classic products of this sort of view. Okay, you have a predefined static sort of structure. These are all the protein interactions in a yeast cell. And so what does that mean? Does that help us understand the cell? What is this that's being described here? Okay, we'll examine that in a bit more detail. But this is the sort of idea. The genome has a blueprint that establishes the structure of a cell. And this is one of the possible representations of this structure that follows straight from the genome and the action of the genes. So 4 characteristics of machines are particularly relevant to the cell and for this metaphor. So the first is that we can describe the cells in terms of a list of parts. That's why we do genetics and how they fit together. That's why we do genomics. And this is, of course, the first part of our dynamic mechanistic approach, where we have to decompose the system before we can recompose it. The operations of a machine are tightly constrained by its function. This is why we can understand them. We can understand them in light of what they are for, what they contribute to the system. So this is a sort of a notion of machine design. So you can predict of, you know, what something does based on, because you know its function, you know what a ribosome does. Okay, so you can look at its structure and you can connect that very easily. So that's the idea here. Machines are highly efficient at what they do. Always follow the same sequence of steps in every cycle of their operation. So basically, if we understand the sort of, you know, the program, that's where the genetic program comes from, this idea that there is a sort of a sequence of operations in the cell that we can reconstruct and understand. So, and also it does this robustly. So the genetic program, I have endless discussions with some of my colleagues that say there has to be a program, otherwise there is no robust, you know, reliable reproduction of development across generation. That's wrong, okay? We're gonna come back to that when I talk about the inheritance of attractors, okay? 4th point, the operation of a machine is not continuous. We can interrupt it and examine machine parts without jeopardizing structural integrity. So we can take the cell apart, look at the parts, and recompose them at our leisure, right? So we can sort of use the same approach we would to study a machine to study a cell, okay? We're gonna look at these 4 points again at the end of the lecture. But in the meantime, Dan notices in his paper that the MCC, the machine conception of the cell, offers a poor and rather misleading representation of biological reality, because there's 2 main problems here. One, cells are self-organizing fluid systems. I'll give you examples that maintain themselves in a state far from thermodynamic equilibrium by continuously exchanging matter and energy with their surroundings. They're not like machines at all in that way. And they're very small due to their microscopic size and their molecular constituents. Cells are subject to very different conditions than macroscopic objects like machines. And we have to examine what that actually means in more detail. That everybody knows that, but what does it actually mean for the operation of a cell? So that's what we're going to do for the rest of the lecture. We're going to look at how this affects our attempts at understanding the functioning of a whole cell using the dynamic mechanistic approach that I introduced in previous modules. So to illustrate what this is all about, I use 4 sort of case studies that, again, are taken from Dan's paper. The first is sort of, we're going to look at the architecture of the cell and how it comes about through self-organization. The second point is we're going to look at this idea that protein complexes are molecular machines. They're not. And I'm going to explain what they really are. And we're going to look at a particular example from intracellular transport, motor proteins. Are they machines? And then we're going to very briefly touch upon the stochastic nature of the behavior and the regulatory systems that control the behavior of the cell. Okay, so first, let's look at the architecture of a cell and the role of self-organization. So the traditional view has it, the genetic program view has it, that we have the central dogma, information going from DNA to RNA to polypeptides, the genetic code. This is a straightforward application of information theory. So the DNA instructs the protein, and then the proteins sort of aggregate together by the, following the laws of physics into this structure that's represented in this beautiful drawing of a cell. Okay, so there is a structure that's well defined, compartmentalized, and often depicted as in this sort of static manner here. But the problem is there is a very big gap between genes making proteins. We talked about this when I showed you this gay gene picture, right? I mean, how does the gene actually affect other levels in the organism, like the cellular level here? It's not clear. So a miracle occurs here in this view. There's a large explanatory gap here. And of course, modern cell biology and developmental genetics are sort of trying to fill that gap, but with limited success. Why is that? So one problem is this sort of static, depiction of the cellular structure. It's not static at all. Here's a beautiful image by David Goodsell. He has lots of these very detailed paintings of the molecular structure of cells. And you can see this is a bacterium, a very simple cell, Mycoplasma mycoides. And you can see all the different protein structures in it, and the cell membrane and the cell wall structure around it. And it's a beautiful depiction, but it's very sort of, okay, so there is sort of a scaffold, there is something that is stable here. And so all these metaphors, these architectural metaphors, so there's the architecture of the cell, and there's a blueprint of that in the genome somehow. Okay, so that's the basic idea behind the genetic program metaphor. So this is the picture that Dan shows in his own paper. Electromicrographs for ages have shown this very sort of static, compartmentalized picture. So it's like a factory. There's exchange between different compartments, but the structure is more or less stable, right? Now, we have to point out the difference between two very different, alternative processes. So this genetic program view assumes that proteins, they self-assemble. Self-assembly is just the physical association of molecules into a static equilibrium structure that does not require any external energy sources. So that's, the protein fold into the right sort of conformation. They form complexes, and that forms the cytoskeleton, et cetera, et cetera. That's just physics, okay? But that's not really what's happening in a cell. And what's happening in a cell is called self-organization, and that's very different from self-assembly. Remember this concept of aggregativity that I talked about when I talked about WIMSAT. So the idea there was that an aggregate is just the sum of its parts, while the complexity of a system is measured by how far it is not just the sum of its parts, right? And so self-organization is what's contributing this extra ingredient. And self-organization is sort of collective behavior of non-linearly interacting molecules that generate a far from equilibrium dissipative structure. Okay, so we're going to go into a bit more detail in just a bit. So this is a self-organizing structure here. This is the nucleus and the mitotic spindle during mitotic division. You can see the different phases of the nucleus from prophase to telophase, and it's beautiful. You see the chromosomes in blue and the spindle structures in green. You can see how this is an amazingly dynamic process at the level of the entire division process. But let's focus in on a particular stage. For example, here in metaphase, and look at the spindle at that particular time. Here's a beautiful movie of a spindle that has been labeled with very low concentrations of fluorescent tubulin. So you can see particles being incorporated and sort of moving through the microtubules. This is, if you look at it on a picture, it looks like a stable spindle. And this is how these microtubules are sort of built and rebuilt all over again. So it's a very dynamic structure, even at this very short time scale. Amazing. And what's more, this structure can adapt to cell size changes, all kinds of perturbations in a self-organizing way. This is my favorite example here. I talked about early cleavage divisions and blastoderm divisions in Drosophila. Here you see them. There's waves of divisions going through the embryo. Remember, this embryo is one big cell. And I also told you already in an earlier lecture that if you kill the nuclei, the DNA in this embryo, this goes on as if nothing happened until gastrulation. So this is a self-organizing process across hundreds and thousands of nuclei. Wave of mitosis again here, beautiful. And you can see the spindles very small. So this is one cell, but a much larger time and spatial scale than we just looked at before. Massive amounts of self-organization. We have no idea how this works. Beautiful, okay? Absolutely amazing. So here is good cell again and a picture of the nuclear pore, the internal structure of the nucleus. It's not, so the chromosomes are wobbling around. There's these sort of opening and closing of the chromatin. The chromosomes are tugged around to initiate transcription here, there. It's amazing. It's amazingly complicated and dynamic. So self-organization arises from the collective behavior of non-linearly interacting molecules, generating a far from equilibrium dissipative structure. We had that already. What does that mean? That means that there is no external template, or there is no global plan, no centralized authority. There is no blueprint to do this. Okay, so this is where the missing information comes from partially during development. That's, being added when a cell is self-organizing. It's not necessarily based on information from the genome. The cell is continuously transforming its internal architecture, and that is the most important point here. There is no static structure. It changes all the time. The cell is basically, every moment of its life, it's rebuilding itself. So Dan calls this emergent mechanics. We'll talk about emergence a little bit more when we talk about novelty and evolution, but this is a very important concept. So it's unpredictable. Emergent means unpredictable. So something is emerging over time, and you cannot necessarily predict what's going to happen next. This is very important. Okay, so self-organization is exactly what confers anti-fragility to the system. It enables the cell to respond rapidly and adaptively to external perturbations. I told you about the spindle, how it adapts. And so this is a system that is very flexible, and it learns from assaults, from mistakes, if you want. So these cellular structures, I even put them in scare quotes, are basically, and remember this, stabilized pattern processes. Paul Weiss would be happy if he were still alive. So this is what a cell is. It's a stabilized pattern processes consisting of thousands of sub-processes that are interacting with each other. And their true nature cannot be captured by abstracted, timeless snapshots, such as network graphs, beyond networks. This is where we're going. So the network graph is just sort of an idealized, momentary picture of what's happening. But the structure that's depicted in a network is constantly being rewired, reformed, is never constant. And this is the essence of being alive. We'll talk about that in next lecture. If you don't do this as a living system, you're dead. Okay, so this constant transience, this constant turnover of your own structure is actually a fundamental characteristic of life. So protein complexes. Very sort of widespread idea here is that, here's the car again. You see, a molecular car. We like cars as machines, not only computers. So the idea is the proteins and the complexes they form are seen as molecular machines, so little mechanical contraptions that form the cell. So here are two people who were really adamantly, or are still adamantly proposing this. Sidney Brenner famously, and then Bruce Alberts longtime director of the National Academy of Sciences in the US and author of a very influential textbook on cell biology, writes, "The cell is a factory that contains an elaborate network of interlocking assembly lines, each of which is composed of a set of large protein machines." Okay, so that's the view. And of course, the quintessential assembly line is the ribosome here, shown in a little GIF animation, which is sort of, incorporating tRNAs and incorporating the amino acids into the protein. So this approach of molecular biology addresses both the aspects of functional decomposition. So you identify the structure of the component parts and assign functional specificity to those structures. Okay, so the proteins have a certain conformation, the complexes have a certain composition, and that tells you what they do. Most beautiful and most famous sort of achievement in this sense is, of course, the double helix of the DNA, which explains immediately a mechanism of heredity, as famously said in the last sentence of that paper by Watson and Crick in 1953. Okay, but there are a few buts here, okay? One is the proteins don't actually assume a unique 3D conformation in the cell at all, okay? They behave more like dense liquids or melted solids. And how they behave is determined, and this is a very important concept here, by the cellular milieu. So the cellular milieu provides the context in which this all happens. Now, the cellular milieu is a function of the entire cell. No less than everything is important to determine the cellular milieu. So we'll come back to that problem in a future lecture, but keep that in mind. Rapid switching back and forth between a range of low energy conformations. That's what proteins do. You see a little movie above there. Some proteins don't have a stable structure at all. So one of the most amazing discoveries over the last decades was the presence, actually quite high frequency, of intrinsically disordered proteins. And these proteins, they don't have a specific structure. They have a very sort of loose conformation. And the point is that the very lack of specific structure is often essential for their function. So it's very important that they don't have a rigid conformation that they fall into. And also the other aspect is protein promiscuity. Here you see an example. There's a specialist enzyme that's only catalyzing one reaction. A promiscuous enzyme is catalyzing many reactions, but it could also be structural proteins doing different things. Beta-catenin is an example of structural protein that acts in signal transduction as well and transcriptional regulation. So functional promiscuity of proteins is more widespread than previously thought. The more we know about proteins, the more we find out that they are involved in all kinds of different processes. Protein function is extremely context dependent. Again, the cellular milieu, the overall milieu that is provided by the entire cell is important here. Molecular associations are stochastic and short-lived. They come and go because they're mediated by very weak van der Waals forces, generating very low activity. So this is a very noisy environment. Everything is happening and associating, disassociating. And that's not a bug of the system, that's a feature. It's very important for the cell and it exploits that. We'll come back to that. Proteins, therefore, Dan writes, are pleomorphic assemblies, not molecular machines. So pleomorphic means they have a huge variability over conformations and over time. And it's very fluid and dynamic what's happening there. So there's no rigid structure, okay? They're very sort of hard to grasp. There is an argument to be made that proteins also are processes, of course. So let's move on to the next point because we have to wrap up the lecture very briefly. Intracellular transport. I just want to mention this because, here's a kinesin walking along a microtubule. And this is sort of the quintessential machine protein, right? It's a motor protein. It's dragging a vesicle here. And this is a beautiful movie. It's called The Inner Life of a Cell. This is just a small part of it. I recommend you watch it. It's beautiful. But is it real? Okay, so this movie, beautiful animation, reflects what's called the power stroke model of the kinesin. Basically, the kinesin burns ATP and so cycles through a number of rigid conformations to create this walking movement here that we see, these cute little feet attaching and walking along the microtubule. Is this really what's working? what's happening in the cell. And there are several problems. It doesn't work at all that way. So one problem is that the protein structure is simply not rigid enough to allow the cycling through these specific conformations. So it couldn't drag this huge vesicle along it. It's physically impossible. So there's constant thermal and quantum fluctuations that disturb its sort of process. It would be like swimming through molasses or walking through a hurricane. Life at low Reynolds number, at high Brownian motion, at a small scale, is very different. It's very viscous. And it's not dominated by gravity, but by molecular interaction. So we have to take this into account. So the power stroke model doesn't really work. There's an alternative model of how these motor protein works, and it's very different. So basically, it's called the Brownian ratchet, and it uses Brownian motion for the motor to advance. So basically, you have a kinesin molecule or a dynein or some other motor protein, and it is changing randomly conformations, okay? So it is going through these sort of different motions that you see in the movie, but not in the right order. And so what the burning of ATP, the hydrolysis of ATP is doing, it's imposing this sort of ratchet structure. So you're randomly moving around, but there's a bias. So if you go over a peak like this, you go further down and then the next step and the next step. And experimental evidence shows that these proteins are actually covering quite different distances with each step. I mean, there's a lot of evidence that's really consistent with this ratchet model. So basically, this is fascinating. Then the next step, it's noisy again. It's sort of doing stuff randomly, but by burning the ATP, it's sort of imposing this energy landscape. So it's biased towards certain conformations that move it in a certain direction. So basically, this is very different from the machine here, because there is no sort of well-defined sequence of steps, but the protein is moving anyway, and it's using the Brownian motion as energy source. So it's not just using ATP. So it's not moving because of in spite of the stochastic fluctuations, it's moving because of the stochastic fluctuations. The cell is harvesting what we call noise, these fluctuations at the molecular level. Last point, very briefly, because we're going to discuss this a little more in future lecture. Here you have an amoeba. It's moving. Beautiful movie. And it's moving with purpose. We're going to come back to that as well. It's going somewhere. It has an aim. I'm not saying it's thinking or conscious or anything like that. It has agency. We'll talk about that. So how does it create this sort of goal-oriented behavior? It creates it out of fundamentally stochastic processes. Okay, its behavior depends on its internal regulatory state, on signal transduction and transcriptional responses. Now, these responses are stochastic as well. So you can imagine two models. We've encountered a French flag model where cells interpret the concentration of a gradient and then switch on certain genes. So basically, in this model, in this view of the world, there's a graded response. The strength of the inducer that, here you have a petri dish with different cells, you have a signal that you apply, and you increase the strength of the signal. So in this model, the response will gradually change across the population of cells, and they will show stronger and stronger responses as you increase in the concentration of the inducer. But what's really happening, we know now, is that there's a stochastic response. So some cells will switch at low levels, and some cells will switch quite discreetly at higher levels of concentration. So this is a stochastic response, okay? And it turns out that signaling and transcriptional responses, again, harvest fluctuation stochasticity in a certain way. They use it, okay? And that's one reason for this is because transcription is intrinsically stochastic. So we've learned recently that through beautiful experimental work that transcription happens in bursts, okay? So what we thought before is that there's a certain level, the rate of transcription is determined by a certain rate of random transcription events that are spaced using a random distribution of Poisson sort of process for the technical term. But what's really happening are these very irregular sort of bursts of activity, and then the gene stops again, okay? And they're very fast and they go away. So they produce RNA, and then this is because all the molecular events that underlie transcription are very, very, very fluctuating stochastic. And so it's very unpredictable, and you can't really talk about a stable average rate of transcription. So, and that also means that genes have a certain probability of being active at a specific time or not. But it's really hard and maybe also not really useful to talk about whether they are or not, because in the end, the presence of the concentration of the RNA is averaging over time in both cases, right? So the nature of the cell is fundamentally stochastic, and it's not noise. The stochastic fluctuations are not tolerated by the cell, but they're used by the cell. It exploits fluctuations. It exists on fluctuations and turns them into coherent behavior. So it's fundamentally unlike any machine we've ever built in doing that. So it doesn't work according to a program. It doesn't work according to deterministic principles at all. So to come back to these sort of 4 main points of the machine conception of the cell. The first, description in terms of a list of parts and how they fit together. This is the basis for mechanistic decomposition. So there is no predetermined blueprint or design with self-organization. So getting the parts list and their activities won't help you, because the self-organizing emergent mechanics depends on the milieu of the whole cell, remember? So it depends on the state of the whole system. The operations of a machine are tightly constrained by its function. So we can talk about machine design and we can predict and control the machine, but the cell's operations are not tightly constrained by structure and specificity of its parts. These protein complexes are very promiscuous and very fluid. So it's impossible to localize function precisely. That's a real problem. Machines are highly efficient. They follow a robust sequence of steps, but the cell's operations do not follow any predefined sequences of steps. What a cell does always depends radically on context. Again, the state of the whole system, its environment as well, on everything, okay? And so the last point was that you could take the system apart and study it and then put it together again, but cells cannot be broken down into parts without jeopardizing structure and integrity. So if you stop the system, you cannot just restart it. Life has to continue all the time. So basically, cells do not naturally yield to reductionist decomposition, okay? And so they don't naturally yield to mechanistic explanation at all. That's one problem, okay? So they do not behave in a predictable, deterministic manner. We saw one side of the problem. In the next lecture, we're going to see another side of the problem that affects the dynamical modeling step, okay? And that has to do with the overall organization of a cell or any living organism. So to wrap up this lecture, Dan Nicholson writes at the end of his paper, Monod was wrong. Okay, I agree completely. The cell is not a machine, sorry for the typo, but something altogether different, something more interesting, and yet also more unruly. It's fascinating. We have to look at what it is really. We have to cast away this sort of oversimplified image of the cell as a machine. It is a bounded, self-maintaining, steady-state organization of interconnected and interdependent processes, an integrated, dynamically stable, multi-scale system of conjugated fluxes, collectively displaced from thermodynamic equilibrium. Wow. Yes, so we spent the next 2 lectures, the next one actually, looking at the structural, the organization of the cell, the relations between parts of a cell, and why they're so special. And then in the lecture after that, we look at this dynamic that brings this organization, this structure, into place, that stabilizes it, yet changes it at every moment in time. This was a bit over length, I'm sorry, but it's a very interesting paper. Do read Dan Nicholson's paper, Journal of Theoretical Biology. Here's the reference. And I hope you tune in next time when we talk about the organization of organisms. Thank you very much for listening. Goodbye now.